Hello everyone, so we are going to be going over again some things from my class, and this is going to be microbe metabolism, specifically looking at the metabolism of bacteria. And so, again, this is just for reference later, so there's a test coming up, so I'm making these in review for that. So first, we have to talk about some theories of fermentation, but since this is one of those marked slides, we're just going to breeze through it very briefly. So, theories of how the bacteria actually used up different materials to ferment. So, like, how were these bacteria creating all these alcohols or acids and whatnot? So, chemical theory would be from von Liebig, and he thought that it was a chemical process which was catalyzed by yeast byproducts. On the biological side, we have de Latour and Schwann, and they believe that a sugar fungus or yeast will actually use the sugar and nitrogen for growth. And then unused substances by yeast are then converted into our alcohol. Pasteur, Louis Pasteur, again, we've talked about Louis Pasteur in class multiple times. It is a microbiome class, so we are going to be mentioning Louis Pasteur a lot. He kind of shows a mixture of both the chemical and biological theories. He says that the fermentation of the sugar solution will have these criteria. Okay, so we need live yeast, and it only happens when they're utilizing, again, the nitrogen and other products. Okay, so afterwards, now this is the part where we are going to have to use the icon and actually pay attention here in the slides, because there is one of those icons for study. It's kind of a little study hint. Okay, so enzymes. Still talking about enzymes in the relation to the yeast and also fermentation. Buchner, whose name sounds familiar to the types of funnels, a Buchner funnel. He does some experiments where he has a yeast extract for medicine purposes. He tries to preserve the extract with sugar, but he found it fermented. And he believed that there are agents that were in the extract that were causing fermentation. And these, of course, were enzymes. This is where biochem is born. This is the birth of biochem. Pasteur and Liebig were both partially right, as the yeast fermented sugars and the enzymes were actually responsible for reactions. So, they are both right in Pasteur and the chemical sense from von Liebig. Alright, let's go into the actual metabolism of microbes. So, to talk about metabolism in general, the definition would be the sum total of all biochem reactions in a cell. And of course, we have catabolism and anabolism. Anabolism is putting together macromolecules. And of course, this requires ATP. In catabolism, we are actually breaking down compounds. Therefore, this will release ATP, or the energy that is captured in ATP. All right. Energy, again, the capacity to do work. We have potential and kinetic. Potential is stored. Kinetic is movement or motion. Basics from a physics class or other chem classes. And again, laws of thermodynamics: can't create or destroy. However, we can transform. So like energy, we could transform others' lives for the better. So do what you can to help others out. All right, energy sources. Of course, we have photosynthesis. So we're going to be talking about light as a source. This is for photosynthesis. That's the energy source, but we also need CO2 as a carbon source. The kinetic energy of photons, sorry about the spelling error, will be converted to potential in the form of chem bonds. We also have this term here, chemoorganotrophs. And these get energy from organic compounds. These need photosynthetic organisms. And then sometimes we'll also need chemolithoautotrophs. Chemolithoautotrophs are ones that get energy from organic compounds. However, they make their own food. Very interesting source of life and nutrition. Chemolithoautotrophs. Alright, so now we could talk about exergonic and endergonic reactions. Again, review from other chem classes and other bio classes. Exergonic, think about the starting products have more free energy, so delta G, free energy, than the products. And then this is release. We're releasing the energy. Endergonic, we have a higher delta G in the products this time. And then this will be reactions that require energy. There's a few more definitions. All right, we could talk about the metabolic pathways. So pathways are a series of chem reactions that convert starting products or starting compounds into an end product. Right, so we have linear, which is A to B to C in a straight path. We also have branched, which is like A goes to B or to C, keyword being or. Then we also have cyclical, where the cycle of A to B to C will actually regenerate that A, that starting compound is regenerated. Alrighty, let's talk about enzymes more. So enzymes are, again, biological catalysts. Usually they are proteins, but there's also ribozymes. They increase the reaction rate, and this means the conversion of substrate to product. How they do this is they lower the activation energy, or the energy needed to start the reaction. So there's high specificity between the enzyme and the substrate. So the enzyme substrate, there's high specificity there. Reactions can, however, occur without enzymes, but they're going to happen really slowly. So if we have this thermodynamic chart, we have reaction progress and then energy here, so delta G. And let's say this line will be the changes in energy, so this is reactants and this is products. Now if we have this blue line, if we had an enzyme, we're going to lower that activation energy. So this is with an enzyme. We lowered it by a significant amount compared to when we didn't have the enzyme present. Once again, most enzymes are proteins, and they're going to end with ACE, like that. And there's going to be ones like D-carboxylase. And then the name just basically tells you what it does. So the substrate, or the reactant, again, will make the enzyme substrate complex, and then we'll have our product. Most enzymes will have an active site that will be the target of the substrate. They also have allosteric sites, but let's focus on the active site first. And this allows for substrates weak binding effects to take place here. And this will cause a conformation change. To create induced fit. The substrate bonds are then going to destabilize, and then enzyme bonds will be formed. So new bonds are going to then be formed. Once again, enzymes aren't used up. They could be recycled. The substrate and the enzyme can dissociate from each other, and then you could use that enzyme again. Some enzymes need cofactors. These are the assistants. So we're going to just write. Alright, so if I have enzyme A, let's say our cofactor will be this green thing called B. So that's our cofactor. And if our substrate is C, but it looks like this, normally A and C will not mix. However, when there's a cofactor here, then it could fit onto the enzyme. The enzyme could do its job. Basically, again, a cofactor is an assistant to the enzyme. We also have inorganic cofactors. Those are going to be metals. However, organic cofactors are going to be called coenzymes. Some coenzymes include FAD, NAD+, and NADP+, of course. We'll get to those in a bit. Those are going to be important in our next topic, the different modes of metabolism, so like glycolysis, Krebs cycle, which I'll go over in a very brief fashion since the professor isn't asking us specific steps. It's not Biochem 2, this is still just microbiome. We have environmental factors that do affect enzymes to go over, so there's a narrow range of optimums. Okay, so temperature, we would have a graph like this, and this ginormous decrease would be after the optimum temperature. So at 10 degrees Celsius will double the speed of reactions with the enzyme. And then proteins do denature at higher levels. That's why we see this steep decline in the enzyme efficiency. Alright, we also have to look at pH. Usually neutral pH is good, so that means around 7, going to see that plateau, and then afterwards there's not going to be much we could do afterwards. Okay, and salt concentration. It's basically the same graph of the pH, but usually we want low salt. And again, a neutral pH to keep our enzymes in the best optimum. We could also inhibit enzymes as discussed in other bio classes. We have competitive inhibitors, and these go to the active site. Similar in chemical structure to the substrate. And then they're concentration dependent. Since they are concentration dependent, they could effectively block the substrate. For example, sulfa drugs block folic acid. And when folic acid decreases, that decreases, growth will also decrease. We also have non-competitive. This is where the allosteric site comes into play. Allosteric inhibitors are one example. The action here is reversible. They are again going to bind somewhere else in the active. As the name applies, the allosteric inhibitor is going to go to the allosteric site. There are some non-reversible non-competitive inhibitors. These include mercury, and this reacts with sulfur amino acids. There's also the other property of mercury where it will change the enzyme's shape and make the enzyme non-functional. This will cause the pathway to stop. A regulatory molecule could also be a factor in regulating enzymes, where the allosteric inhibitor will bind to that allosteric site, and the enzyme will be have a distorted shape like that, and the regulatory molecule will then be made afterwards, and then this will make more allosteric inhibitors. So this allows for feedback inhibition. Alright, let's talk about ATP. I know this is going to be quite a long video, but I'm trying to get this whole chapter in one so we could possibly save more time. Alright, so ATP, adenosine triphosphate, energy currency. Alright, so ADP, which is adenosine diphosphate, accepts free energy. And how to make ATP, so you add inorganic phosphate plus ADP. And energy is released when ATP is hydrolyzed into ADP and that phosphate. Substrate level oxidative and photophosphorylation generate ATP. So substrate level phosphorylation is exergonic. Oxidative is proton motive force. And photophosphorylation is usage of light for proton motive force. So those are three processes to create ATP. Alright, now metabolic pathways. Again, looking at the pathways and other parts of them. So, of course, hydrogen. Extremely important. It's a proton and an electron. One proton plus one electron. No neutrons here, just proton and electron. So, reduction and oxidation will come into play in these reactions, of course. Reduction is gain electron, and oxidation is lose electrons. So think of it reducing electrons, or reducing, let me rewrite that. Sorry about that. So lose a proton, or a positive charge. So you're reducing it when you gain a negative, or when you lose a positive, you're reducing it. I think this is a much better way of remembering it. Reduction, you're losing a positive, you're losing a proton. Sorry about the mishap there. Having a brain fart is trying to process these two things. But reduction, you're losing a positive. Oxidation, you're losing a negative. You're losing an electron in oxidation. Alrighty. So the H will actually move. How this will be shown, since that's going to represent our electron or our proton. So we're going to have X plus Y. X will have an electron move to Y, so then X will become positive and Y will become negative. Y is reduced, X is oxidized. Again, Y technically also lost a positive, so since it became negative, that's reduced. Alright, so we're just going to talk about energy sources again. and terminal electron acceptors. So energy sources include organic and inorganics. And then terminal electron acceptors, which are important in our pathway, are going to be O2, other molecules, and the electrons move through a series of oxidation reduction reactions. Alright, so let's talk about those electron carriers. Now we're getting even closer to the big picture, which is all that glycolysis and all those pathways. So, electron carriers. These are going to be reducing power. Again, reducing is when we gain a negative, we gain electrons. And these transfer electrons to chemicals with higher affinity for those electrons. So some electron carriers are, of course, NAD. So this is the oxidized side. So we're going to put an O, and then on this side will be the reduced form, NADH. Oxidized version, NADP. Reduced, NADPH. And then our last one, FAD, when it's reduced, becomes FADH2. So again, oxidized, we are going to, if we go back up here, look for it, we are going to lose an electron, so these actually have a little plus sign by them. We're going to lose electrons here, however, we're going to gain electrons here on the reduced side. We also have precursor metabolites. So, intermediates of catabolism can be used in anabolism. So, an example of this are the carbon skeletons. Again, it's almost Halloween. And these could be used for building other macromolecules. That's all we would need to say there, but in Biochem 2 and other classes like that, you could go into much more deeper of a dive into all that. Okay, so overview of catabolism. Now we're just switching gears, going into the actual parts of the pathways. So catabolism. Again, this is breaking down. Okay, so we have the central metabolic pathways. These include glycolysis, the pentose phosphate, and the tricarboxylic acid cycle. So those three types. The outcomes are ATP, reducing power. Remember, that is gain a positive. I think that's what I put. Or gain an electron back here. I kept switching it around. I'm sorry. Reducing is, of course, gain an electron, so gain electron. Sorry, I keep mixing that up because I keep referring back to something else. Sorry about that, but reducing power again, gain an electron. And we also get precursors. All right. Respiration. Let's talk about respiration. Breathe in, breathe out. That's also part of respiration, but in this field, we're going to be calling it the transfer of these electrons from glucose to the electron transport chain. And the ETC generates proton motive force. And this is harvested to make ATP via oxidative phosphorylation. We have, of course, aerobic and anaerobic. I'm going to just put one of these here. So O2 is the terminal electron acceptor here, while other molecules are the terminal electron acceptor in anaerobic processes. Fermentation is therefore so you highlight EDC and fermentation, there we go. And this is where organic molecules are the electron acceptor. And they regen NAD+. Remember, that's our oxidized form. NAD+ is oxidized. I'm going to just write that here. And pyruvate is a very common one as the terminal electron acceptor. So again, fermentation, organic molecules are the electron acceptor, and you use that to regenerate NAD+, which is one of those carriers, but it's NAD+, so it's the oxidized form. It lost an electron. Glycolysis. There's so many steps in glycolysis, and those are going to be fun for me to go through during Biochem 2 when I'm making these videos for Biochem 2. But I even spelt it wrong, so that's kind of funny. Alright, sorry about that. Okay, so in Biochem 2, we are required to know all the steps and detail the enzymes as well as the products. But since this is microbiome and not Biochem 2, which I'm taking next semester in the spring, we're just going to go through the basics. So glycolysis, we're going to convert one glucose into two pyruvates. Again, each of these have 3 carbons. And this net yield of 2 ATP and 2 NADH. That is the reduced form. Alright, the investment phase. Will have us use 2 ATP, and this adds 2 POs from the 2 ATP, and glucose again becomes pyruvate and pyruvate, which have 3 carbons each. This glucose has 6 carbons. We also have the payoff. Again, there are so many enzymes and so many other processes that happen here. And that's, again, for Biochem 2, not Microbiome. So we'll be going over those possibly in the future when I take that class. Alright, so 3 carbon molecules are converted to pyruvate. This makes 4 So makes 4 ATP and 2 NADH. And again, our net is actually 2 because we spent 2 here. We spent 2, but we made 4, so our net is actually just 2. No oxygen here. Alrighty, let's go on to the next step, the pentose phosphate pathway. This is a small section. There's not an icon here, so I'm going to breeze through it. Breaks glucose, and it creates these precursors like ribose 5-phosphate and ethyrose 4-phosphate. It creates NADPH, reducing power. And again, this is the, let's go back up to our chart. We gained an electron since it's a reducing power. And then it also has varying yields, but that's the run through there. Again, it's not a big part, so we're going to just move to the next major step here, the transition step. This links glycolysis to the TCA cycle. Here, we're going to remove CO2 from pyruvate. Again, an enzyme, probably a decarboxylase, something like that, will remove that CO2 there for us. Electrons transfer to NAD+, and this becomes reduced to NADH. Again, reducing power because we're gaining an electron. Acetyl-CoA is made when CoA is added. And this takes place in 2 different areas depending on what type of organism you are. So the transition step in the prokaryotes will take place in the cytoplasm, while in eukaryotes it happens in those mitochondria. The once free living bacteria. Alright, now let's go into the TCA or the Krebs cycle. This is the big one. Oh yeah, Mr. Krebs, right? Alright, so this completes the oxidation of glucose. So again, oxidation is the loss of the electron. And this produces 2 CO2. We also have 2 ATP. We have NADH. We have 6 of those. We have FADH as well, and these are made, there's 2 of those. We also make precursors. In my actual notes, I wrote a ton of things down for glycolysis. I'm just going to say them very briefly here, just in case. Alright, so 2 pyruvate, a bunch of phosphorylations are happening here. So PO-lation. To get more to those 4 ATP, we have a lot of phosphorylations constantly throughout. Alright, in the TCA cycle, I actually wrote a ton of things here in my actual notes. I wrote, we have to take away one of the carbons, so -1 carbon. This is probably in order. the teacher was going really fast through them. So -1 carbon from the previous molecule, and that creates CO2. And then we have reducing power again made, so one of our NADHs is probably made there. Now we have acetyl-CoA. Plus the oxaloacetate. That's gonna be fun. We'll create our citric acid, and then we take another carbon somewhere, not 7, one of them, and this becomes alpha something. I couldn't really catch it when the professor is going over it really quickly. Again, he doesn't want us to memorize the actual steps for it, because again, this is not Biochem 2. This is just microbio. And we have other steps, a lot of phosphorylations, more reducing power, forming that CO2 again. NADH is made, and precursors as well. That's what I wrote there. Very interesting story of Hans Krebs and what was happening in Germany at the time. He moved to Britain, and that's where he was able to do all the experiments to figure out the Krebs cycle. Alright, now the electron transport chain in the prokaryote. Let's talk about the ETC. Did I highlight that? I did not. Completes oxidation and then all of those. Okay, the ETC in prokaryotes. Sorry, it's so zoomed out. There we go. Alright, let me see what I wrote here. So, these are membrane electron carriers. So they eject protons from high to low concentration of those protons. The eukaryotes have this happening in the inner mitochondria membrane. There's an electrochemical gradient that forms. And this is used for ATP. And it powers transport. Such as flagella. And I wrote NADH is just the start of this process. Yes, in the chart that's there, which I'm not going to copy because there's so many things to write for it. NADH is the starting part. Again, that's probably going to be going to depth in Biochem 2 when I take it. I make a video for that, but right now it's just the basics. We also have things called quinones, cytochromes, and flavoproteins. And just like a Starbucks, label, I think I butchered all those names by accident there, so sorry about that if the pronunciation got really messed up there. I did write the steps of them, so I'm just going to read them out. So NADH has 1 proton removed, and this is done by a dehydrogenase. Again, ACE, it's an enzyme, dehydrogenase. That means, I said it there weirdly, just to say it removes the hydrogen, dehydrogen. Okay, something called ubiquinol. I butchered that. NOL is at the end. NOL, I think. And then this removes another proton. This makes proton motive force. We got some force here. Alright, ATP synthetase. Once we have that proton motive force. this is where the hydrogens will enter, and this will be driving phosphorylation as the hydrogens enter through active transport. So this means a low to a high. This also powers, again, the flagella are powered because the activity of the electron transport chain in the prokaryotes. This is what powers the flagella. And now the reducing power of glycolysis and the Krebs cycle now has a use. This is the eureka moment. Alright, let's talk again about the ATP synthase. This is 1 ATP is made for every 3 protons. Okay, so the next part, of course, and this harvest proton motive force. Alright, ATP yield, so the big picture overall. I'm just going to read the charts as substrate level and 2 net ATP from glycolysis, 2 ATP from the TCA cycle. So this is 4. Actually, I should write it up here. Oxidative phosphorylation, so 6. So this is from reducing power in glycolysis, or made from glycolysis, I should say. It's gained from glycolysis, this reducing power. And another 6. So this is reducing power gained in the transition. And then 22 ATP from the reducing power gained in the TCA cycle. Right, so that means a 34 total here. And then total ATP gain. Would be 38. And then this is a theoretical. Alrighty, so that's basically all of those steps. Now, we could talk about fermentation briefly again. That's where we all started this. Alright, so this is used when respiration isn't an option. And organic molecule, again, an organic molecule is used as the final electron acceptor. Okay, so let's talk about this in context of microbiology. E. coli bacteria is a facultative. Anaerobe, meaning it can survive in oxygen conditions as well as non-oxygen conditions, so it could do both aerobic and anaerobic respiration, as well as fermentation, if it likes to. So the pneumonia bacteria, Streptococcus pneumoniae, this one does not have an electron transport chain, so it must only do fermentation. Okay, so fermentation, again, it doesn't generate ATP, only glycolysis gens ATP. That's kind of an important note, so we're going to highlight that. Organic molecule is a terminal electron acceptor. We're going to also highlight that. Additional steps. Why did I spell that? Oh my goodness. Will consume extra reducing power to make our oxygen or oxidized form NAD plus again. Alright, and a lot of things are made by fermentation. We have dairy, pickles, a lot of other things, ethanol, of course. Gulp, 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 you know. Butanol, another alcohol, acetic acid, in cheese, also lactic acid. A lot of things are made in fermentation. Use of other substances. Since microbes are able to use a ton of other forms and other molecules for metabolism, so they could actually excrete hydrolytic enzymes or enzymes that perform hydrolysis, and then they transport subunits in once they're finished breaking it down to degrade it further. And then again, that makes precursors. So polysaccharides and disaccharides. And so amylase will digest starch. And then, cellulase will make cellulose, or digest cellulose. And then there are specific enzymes that hydrolyze disaccharides. Lipids. How they use lipids. How do bacteria use lipids? So we, of course, have the fats getting attacked by lipases. Example would be glycerol being converted into dihydroxyacetone phosphate, and then this enters glycolysis. Fatty acids are degraded by beta oxidation to enter the TCA cycle. Fatty acids, TCA, fats go to this step here by lipases and then to glycolysis. Proteins could also be metabolized, so hydrolyzed by proteases. I'm just going to highlight proteases. I should get the point. So amino acids are then deaminated. So they take away the nitrogen groups on there. And then again, the carbon skeletons, the spooky, scary skeletons of the biochem world, are then turned into precursors for other usage. And then our final portion is on those chemolithotropes. So they use reduced inorganics as sources of energy. So things like hydrogen sulfide and ammonia. They're made by anaerobic respiration, and then sulfate and then nitrate are the TEAs, terminal electron acceptors. And then this is nutrient cycling, which is extremely important for the environment. So there, I'm going to put a big star there because how important the nutrient cycles are for the ecosystem as a whole. Alrighty, so that's just a rundown of bacterial metabolism. Thanks so much for tuning in, and please do something nice for someone. Best of luck.